Thank you, ma'am. Thank you, AIOS and the Ajanta for organizing this. My talk is about cells, COVID care, and personalized medicine. These are my financial interests. So one of the things we did last year was publish a few papers on COVID-related work. And one of this was a hypothetical-based review of COVID prophylaxis for healthcare professionals. We use this concept because of Trehalose, because we found out from the other parts of the body, from the medicine, it does a lot of antiviral activities. And because it had a lot of antiviral activities itself, we thought that why not use this as one of the areas of prophylaxis. I'll just go into this here. And this is how a virus enters. This is a paper in the early part of the epidemic, I think it was published in June or July, very early. And it was a very interesting paper about how the COVID cells enters through the ACE2 receptors. I want you to remember this picture for in your mind because I'm going to use a lot of things related to this image over time when I keep speaking. And there is a pathway where Catapsin induced endocytosis, which is a very important thing. That's how the virus enters. It binds to the receptors, use this particular enzyme, and then it gets into the cell, and then it starts replicating, creating your RNAs, and then all that it controls your body. This can happen in any cell. It can happen in your cornea. It can happen in any part of your body or lung epithelial cells, depending on the receptors which it needs to enter. So there are two important things at that point of time, the chloroquine and Trehalose, because something it was easily available. And you can see here that in many parts, it reduces the catapsin pathway. It reduces this related pathway. This is what we believe in. And also it helps in, degrade the enzymes. And all this helps in reducing the viral load and doesn't allow it to replicate at all. And this is a picture about how chloroquine and both Trehalose work on the epithelial cells. This is nothing to do with COVID, but in a regular dry eye kind of a picture here. So the first question, that was the first thing, which was more of an hypothesis at that point of time. We didn't have access to the COVID eyeballs. Unfortunately, the second wave got in a lot of new challenges. One of the challenges was the eye bank challenge. We used to get eyes from patients who had no history of COVID but had a silent MI, and the physicians had not done COVID tests because it just looked like MI. However, we are one of the few eye banks in the country where we used to take the swab, and having our own COVID testing laboratory, we could come and get the swab of this test person for the checking of COVID status. And we would keep the eyes for 24 to 48 hours till the report comes. And we were surprised, out of say 100 eyes we collected, close to 20% of them were unknown COVID positive patients. And if we had not done the COVID testing, these corneas would have gone to somebody and they would start using it. However, with the ethics committee clearance and all the other regulatory body clearance, we thought, why not use this COVID eye to see how the gene expression, how the response of the ACE2 receptors, everything we wanted to study at the microcellular level. These are one of the very, very few there's not much of work that really happened on this project yet. And we are one of the few and fortunate to be able to do this, to come to the scientific community, what we really plan to do with this data. And we looked at gene expressions, we looked at the receptors, which helps to enter the eyes. I'm not going to too deep into this. Look at these ACE2 receptors, IMPRES, CTL, all these catapsin-based pathways. These are all important ones. These are in the healthy eyes. You can see those green ones. These are all your receptors. It's all labeled. Also, we looked at from cornea, conjunctiva, trabecular meshwork, choroid, and each and every layer. At this point of time, my colleagues, Dr. Sharon, Direct, Pooja, and Purnachandra Tirumalesh. All of them were very, very instrumental in this whole project. It's one of the very, very important projects because dissecting it out, getting these eyes, trying to get this organized and sending it immediately to the lab for all the staining was very important. So what we found was, this was a control eye. We found that the retina did not show much of ACE2 receptors. So that means the chance of virus going directly into the retina and attaching itself was very low. The conjunctiva and cornea, especially the ocular surface, had a very high level of these receptors, something similar to what you see in lungs, which means that we are filled with it. And you can see that these red are those receptors. And we also stain for the antiviral staining. And these viral interferons, these interferons are the ones who actually kill or reduce the viral load, which you see completely is absent in a COVID positive eye. And in the COVID recovered eye, it's relatively better. But you can see that the conjunctiva is one area where the receptors are heavy. And even in your cornea also, you can see that it is there, but as not as much as what you see in the conjunctiva. And this is a very important feature of the antiviral activity, which controls the role of how the viral will replicate, but it's completely absent or low in the COVID positive eye. And you can see that there are also which stain with all the thin press and in the stroma and the recovered eyes. They were not the real important factor at this point of time. But when you look at the catapsin, the one which I showed you, this is the one which is catapsin here. If you remember this thing, catapsin-based thing, we also stained for this. And what we see in it is exhibit immense catapsin in epithelium while experience limited in the control and recovered. That means that catapsin, which is the green here, is extremely localized. It's very high in this zone, and you don't see it in the recovered eyes, and you don't see too much of this on a healthy eye. So what is important here is there is a mechanism of the COVID virus entering into the eye, exactly like what we have seen in the picture here, which was at that point of time last year, a diagrammatic picture. This was taken from different, it was adapted from a nature, and we adapted to our localized, to the corneal part of it, the ocular surface part of it, but exactly what we saw a year later with these eyeballs, which is quite interesting and also scary at the point that these cells are changing. Both in the control eyes is different. Of course, we don't expect them. The COVID positive eyes is different. You can see that even in the cornea, and the recovered eyes is completely different because it's changed. It behaves something similar to the control. This is how beautifully the cells are behaving. And these positive eyes are not the patients where we got the eyes from the ICU or we did not touch. They were more of an asymptomatic positive. That means they didn't even know that they were positive. And that is what is very important at this point of time. And we started looking at potential therapeutic targets. We looked at trehalose and chloroquine. And what we found was these were on the HCE12. The trehalose activity increased the antiviral change. I've not put this picture, and this is a very, very important factor out here because antiviral activity is this. I'm sorry, my slide is filled with all these diagrams, but if you look at this in the control eye, and you can, a little bit of this is here, not too much. This is what we need to increase. And that is what increased with the trehalose. And that is why we've been telling, even last year, at this point of time, exactly what does it do? It reduces the catapsin, increases your interferons. And this is what we want to express here with this experiment. This is one of the world's first time we're going to show this, that we're just ready to send it for a Nature journal, one of the important journals, is that this is the increase. This increase, this increase of the interferons is what is very important with trehalose. The second point, Madam has already covered this, but we did some experiment. What she spoke about the mask is very right. It's about the hypercapnia, and hypercapnia changes everything. We did a study with the tears collected before the COVID started and after we put the mask. And I'll just go directly what it happens is the hypercapnia changes the entire ocular surface in a different way. Hypercapnia is carbon dioxide, and carbon dioxide is not supposed to be pumped in all the time. If you wear a mask which is loose, it goes up. And what is important is the mask related dry eye is completely different. And you have patients who complain for very vague symptoms because your molecular pathways are completely vague. And that is why patients who come with mask, even though they say they don't wear mask for a long time, still you have a vague symptom because this can change. Because all the time we are used to looking at hyperosmolality. But what I want to tell you here is mask-induced dry eye induce hypo. Remember this, it's hypo, not hyper, which we are already used. So drugs which work on hyperosmolality dry eye should not probably be used. We should look at drugs which are, I don't even look at, drugs which are the older generation one, which are not a hyperosmolality reducing drugs, is something which would work very well out this year. So what I'm to say is, you can expect delayed wound healing because of hypercapnia and this non-responders to treatment, different type of bacteria, because bacteria which are anaerobic and aerobic are completely different. Because bacteria, you will end up in having bacteria who can survive without oxygen and probably survive with carbon dioxide. Different inflammatory markers, the corneal nerves are changing, and ocular discomfort will be completely different from what we know. And if you are a keratoconus, we're already seeing this. There's a lot of changes in the way keratoconus progress because of the change in the ocular surface milieu. Thank you. Thank you, ma'am. And